All right, so this is how to set up the floating leaf disc lab for photosynthesis. I do this lab in the energy unit. So after we talk about food webs, we get into photosynthesis. Students have already learned about the process of photosynthesis after taking their notes, and so they already know that the oxygen gas comes from the water molecule, and the glucose molecule comes mostly from the carbon dioxide as well as the hydrogens from the water. So I've already dealt with their misconception that plants directly turn carbon dioxide into oxygen gas. They now know that that's incorrect, that was a lie their elementary school teacher taught them. They do take in carbon dioxide and they do release oxygen, but the oxygen is actually coming from the water molecule. So it's actually this whole process of splitting up the water molecule into oxygen that we're going to look at through this lab. Students have also already learned that white light is made up of multiple colors. In fact, we already demonstrated this by using little diffraction gratings that we put over their cameras and stuff, and they realized that all of light is separated into the spectrum of colors that they know as the rainbow. With that knowledge, you can use this experiment to test all sorts of things. How do different colors affect the rate of photosynthesis? Might be something interesting that they can test underneath these lamps using some different filters. For the record, diffraction gratings are just about the coolest things ever to split up light into its different wavelengths. It's a great photosynthesis demonstration for students just to see all the wavelengths that are normally hitting that leaf. So here's the setup for the lab. Each table is going to get one desk lamp as well as 4 beakers and 4 syringes. So each pair gets 2 of each of those. Now, I usually get ivy leaves for this experiment because there's big branches of it that you can kind of pick off and bring into the classroom. So I'll usually get big old branches, as many as I can, and bring those in. So sometimes I'll walk out to the forest and go to a tree. Other times, if I just need a little amount, they're growing right here along the side of BFA. Ivy is also still green in the wintertime, so it's a year-round leaf supply that I can get at any time in the year. Also, just a side note, if you put the green leaves on black paper and then use the diffraction grating to view them, it's you can kind of see how much green light is being reflected off of their surface. You see a little bit of red, a little bit of yellow, a little bit of blue, just a little bit, but you see kind of those bright green colors just radiating off the surface of the leaf. Kind of, if I put my finger up here, my finger's more red, and so you see kind of the red and the yellow outline of my finger in this spectrum that's being reflected. But if you put the leaf up, you see more of the green part of the spectrum. I would recommend setting up this lab maybe the weekend before. So if you do this lab on a Monday, it gives you the weekend to just set everything up before. So just to kind of give you the student's perspective, I'm going to be labeling my beakers, one for the dark experiment and one for the light experiment. So students can get a piece of tape and apply that on in the first period. By the second period, it'll already be on there. The second period will have to take the tape off. Now, different teams will be under the same light, and so it's good to maybe put your initials here as well. Okay, so I'll mark this one with my initials. After students have labeled their beakers, they can go ahead and just fill them up with water to about the top part there. Really, that's all they're gonna, they can fill it up to the top there. Now, the students know that photosynthesis requires two reactants, both water, and they know they have that one, but it also needs carbon dioxide. So where is it going to get the carbon dioxide? Well, that's provided by the baking soda. Okay, so I provide that for the students. The students can just take just a pinch of the baking soda and put it in their water. Really, it's not that much at all. It should start to disperse there. They can kind of swirl it around just a little bit without spilling anything. That baking soda is going to do a chemical reaction that'll produce some carbon dioxide gas that'll be dissolved inside of the water. That's going to help boost photosynthesis. It's going to give it some more dissolved carbon dioxide so that it can do some of the reactions, okay? So students can do that with both of their beakers. Remember to emphasize experimental design with them. So this is a constant, okay? Each experiment has the same amount of water, and it also has baking soda. As you can see, the bicarbonate, the baking soda, creates nice little bubbles of carbon dioxide that get dissolved into the water. Next is the students are going to have to cut up some different leaf discs. And so they need 10 exactly. So students can use their hole puncher to punch out those little discs. They should avoid main veins. Now, to get enough hole punchers, usually I just go to the supply closet and they have a bunch there. Even if it's not this exact type, students can put the leaves underneath the paper hole punch and it works just fine. After you get 10 for the light experiment, we're also going to need 10 for the dark experiment. Okay, so now that I've got my group of 10 little leaves, I'm going to put those discs 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 inside of my syringe here. Okay, so the parts of the syringe, right here I'm holding the barrel, the hub is at the end, and I'm going to put the plunger back into the barrel. Okay, be careful to remind the students not to crush the leaves too much, okay? So we're just gently pressing them up against there. We're not crushing the leaves. All right, so here I have my carbonated water here, and I'm going to take my syringe and I'm going to draw up the water. What you'll notice the problem is that these leaves have already floated to the top, and we want to measure the rate of photosynthesis by them floating to the top, because they float to the top because of air bubbles. Well, they already have air bubbles, and those air bubbles aren't being produced by photosynthesis right now. We're already there. So how do we get rid of those air bubbles? Well, we got to create a vacuum. So in order to create a vacuum, okay, I'm going to push out as much air as I can, and really I already have. And then I'm going to pull on this, okay? And as I put my thumb on the edge of the syringe and I pull back, okay, that is creating a vacuum right there and sucking the air out of the leaves. And what'll happen is they start to float to the bottom. Okay, so they've lost those little air pockets. I might have to do this a couple times to get rid of all of the air. So vacuum it up, and then release, okay? So now all of my leaves have successfully sunk to the bottom. None are at the top. And the only way that they would get to the top is if they got more air bubbles that would come from producing oxygen gas from photosynthesis. Empty those into the beaker once again, okay? And what should happen is the leaves should hopefully sink to the bottom there. So this is my light experiment. What I'm gonna do is I'm gonna place it inside of this photosynthesis light bath that I've created out of a copy paper box. I've cut a hole out of the top here, and I basically illuminated aluminum foil and put that inside of the box. And so when I put the lamp inside, it's going to reflect light all around, and this increases the lux at which the the light that the leaves can receive. So now at their station, students have this little light bath, and they can see if the leaves start floating at the top of the beaker. So students should be measuring that every minute. Students can use timers on their phone. They can be looking through the top of the box. They can be looking through the side of the box. Whichever way they do it, every minute that passes, they should be recording in their graph the number of discs that are floating. Both pairs at the table can place their beakers inside of the photosynthesis light bath and do the experiment at the same time. One thing students should be sure to do is to count the number of leaves that they have at the bottom of the beaker, because sometimes they are on top of each other. So I just realized I made a mistake. These leaf discs right here are actually two different ones that have been stuck together. So that should not have been the case. If I put them back in, that kind of introduces some experimental error there into my experiment. So I don't want that. Students can fill out the graph at the bottom. Have them try to figure out first how to fill it out, but remind them the x-axis is usually the time factor, and then they can try to map out here the number of floating discs over time. So where do we get the setup for the dark experiment? How are we going to put the leaves in the dark? I found the best way to simulate the dark is to use copier boxes as the cover up. So I just come into the supply closet, and when nobody's looking, I just take a bunch of these boxes. After you take out the copy paper from the boxes, I'll just get one of the boxes like this. I don't really need the lid, because what I'm going to do is I'm going to be placing it over the beakers like this, okay? You'll notice that some of these boxes have a wide gap, and I don't want that because I don't want to let light into my dark experiment. So what I'll end up doing is I will just close that, and then I'll use a stapler there to staple those two things together so that I've kind of stapled that shut. So again, for the dark experiment, students are doing the exact same thing. They're loading the 10 of their leaves into the syringe. And so students can do this simultaneously, like if one pair wants to do A and the other pair wants to do B, and I'm going to pull, and it really does suck all the water out of the leaves there. And it's kind of fun to release it like that. Can make sure none get stuck inside of the plunger, so you want to make sure all your leaves make it. Okay, and then even if they're floating at the top, once they get in, make sure that they sink to the bottom like they're doing. All right, so this is my dark experiment. It can be happening at the same time that the light one is. So maybe they can just have a box that they end up putting, sorry, get that lamp out of the way, putting on top. So it is completely dark over that, and then just every minute lift it up, see what it's doing, and then place the dark box on. So you have a dark box and a light box. Usually I don't use the light box for the dark box. Like I don't have them use the same one and then turn it back over again, because then that would take 30 minutes. It would take double the time. So you want both experiments running at the same time. Now that students know how to perform this experiment, they can then use other variables to test. Like if I use a green filter, does green light make the leaves rise to the top as much as red does. So I could use a red filter, a green filter, all sorts of things. They could design different experiments with those. When they're done, I have students pour the water back through a filter so that they can collect the leaves. All right, how's it coming?
It is going. I'm keeping this thing here so I can collect all the little dots so I can release them all into the wild.
Release them into the wild. All right. Stuff. All right, are they sinking to the bottom there, Ken?
Yeah.
Okay.
A
Looks
lot
good.
of mine are sinking, but a few of them are still up top.
Okay. All right. Good. So you got them in. So you're gonna repeat the one for that. Position the lamp so that you can look in without having to move it.
Come on, float! Make me proud.
So why are they floating, Sean?
The leaves are taking in all this water around them. And
Okay.
they're splitting the water, and they're taking in the hydrogen and spitting out the oxygen, which is actually why you can see it every once in a while. It'll like ripple
Yep.
completely on its own,
Very
which is
nice.
really strange, and that's because it's spitting out oxygen.
Caroline?
Come on, Beaver! Go! Go! Make it float! Go!
All right, are they floating? Nice. All right.
Now you guys have 1.5.
All right, so the light.
Still two?
Yeah.
Oh, one's rising.
That's theirs.
Oh, theirs is rising. Okay.
You guys got 2 now.
Woo. All right.
No, it has to be to the brim. Does that have to be to the brim so it's kind of exploding?
Yes. Yeah, the water needs to fly. Go to the complete top. So are any of the ones under in the dark floating?
I got two.
2 are floating, but none of the others are.
Oh wait, this one looks like it's going up.
Okay.
Oh!
So why are these not floating?
Yo, 5 minutes!
Photosynthesis to create air pockets between.
So is photosynthesis happening here or not? It's not. Okay, so there's no air. Okay.
Alright, get ready.
We got a floater.
We do?
We got 1 floater.
10 seconds.
All right, how many are floating?
For me, too. For
Okay.
us,
For her,
only none.
None, only none. Okay, so some are floating, some aren't. So we got the dark versus the light.
We got 8.
All right, are they floating in the light?
No.
They aren't. Oh man. All right, we're only 5 minutes through though. Are these floating in the light?
No.
Not yet.
I like
Check.
So are they floating for you?
Yes!
Why are they floating?
Photosynthesis.
Well, what about photosynthesis?
Oxygen. Oh no, one's going back down!
Oh no! Where's the oxygen coming from?
Oh, our timer went off.
Where's the oxygen coming from?
What?
Why do we
suck? God, no floaters here. The water.
The oxygen. The water.
The water. Oscar's coming from the hydrogen and the oxygen, right? Oscar and Henry.